Today we will discuss about this HBsAg vaccine comparisons with the tumor mutated neoantigen vaccines, which is one of the traditional approach to target cancer cells. So in the previous video we have understood that this HBsAg vaccines seems to be working, and it can help in detecting the cancer cells by the immune system of the patients. So once they have validated their platform is or their vaccine is working, they wanted to compare it with their traditionally available competitors, that is tumor mutated neoantigens, to see whether their performance is better than their mutated neoantigen vaccines or not. So along with these two, they also used the control where they used OVA mutated, where they used OVA expressed cancer cells, and there is this OVA expressed cancer cells is easily recognized by the patient's immune system, and it can be used as a control against these two systems. So how they did their experiment is they treat the mice with their respective vaccines, that is HBsAg vaccines and the tumor mutated neoantigen vaccines 2 times with a gap of 7 days, and after the 7 days of their last treatment, they injected the respective tumors, these tumors that contains this HBsAg surface antigens and this tumor mutated neoantigens and this OVA expressed cancer cells. And after this 10 days, they analyzed the different mice from the different points of view that we will discuss. First, they check the tumor suppressions, and they are able to see that this HBsAg vaccines gives maximum tumor suppressions compared to the other alternatives. And simultaneously, they also check the mature dendritic cells, which is high in the case of this HBsAg treated vaccines compared to the control and this tumor mutated neoantigen vaccines. Thereafter, they did the intratumoral immune cells analysis using flow cytometer and find out that the CD4 and CD8 plus cells are high in the case of this HBsAg vaccines compared to the other cases. That is shown in this panel E and panel F also, and G is the common, seems like the common, which has all the CD4 plus and CD8 plus T cells. And in the panel E and F, it is clear that the CD4 plus and CD8 plus T cells are high in the case of this HBsAg treated mice compared to the tumor mutated neoantigen treated mice or OVA expressed cancer cells treated mice. Then after that, they check the subset of these T cells, that whether the CD4 plus and CD8 plus T cells are able to produce lactic granules such as granzyme and IFN gamma. At the same time, they also checked the PD-1 expressions on the cells, which is a sign of activated T cells. At the same time, they are also known as checkpoint inhibitors, which when interact with their counter ligand will cause suppression of the T cells. And all these 3 categories or subtypes of the cells are high in the case of these HBsAg treated vaccines, that can be seen in this panel G, where this CD4 plus IFN gammas are significantly higher. Similarly, this CD4 plus granzyme T cells are also significantly higher. And same things they have seen for the CD8 plus IFN gamma and CD8 plus granzyme. Along with that, they also check this CD8 plus CD279, which is a marker for PD-1 receptors, which is also high in the case of HBsAg. That is, activated cells seems to be high in the case of HBsAg treated vaccines. Then they check the CCR7 dendritic cells, which is the circulating mature dendritic cells, and which has a high capacity to prime the T cells. and these CCR7 dendritic cells are also found to be high in the case of HBsAg treated vaccines. That can be seen in this panel I. And thereafter they wanted to check whether this immune response, the immune response triggered by this HBsAg expression on the cancer cells, will cause endogenous expressions, will cause endogenous T cells activations or not. In order to check that, they check the IFN gamma spot in the spleen cells, IFN gamma ELISPOT in the spleen cells, in the all three cases, and they are able to see that the IFN gamma ELISPOT in the spleen cells seems to be high for the HBsAg treated vaccines. That can be seen from the image in the panel J, and the quantifications are shown in the panel K. And these results are also consistent with the previous results, where they find that HBsAg performance seems to be better than the traditionally used tumor mutated and neoantigen, and also better than the control. So this in all together indicates that the platform, or the vaccines that are produced by targeting or by expressing this hepatitis B surface antigens on the cancer cells seems to have a better efficacy or better immunogenicity compared to the other traditionally available vaccines. This is what they have validated in their experimental results that are shown in this figure. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.